Hi, my name is Amit Chawla, and today we will be looking into the research between the link between mitochondrial DNA and neurodegenerative disorders. What is mitochondrial DNA? Well, deoxyribonucleic acid, or DNA, is traditionally found within the nucleus as a chromosome and aids with the growth and development of an individual. However, mitochondrial DNA is found within the mitochondria and aids with the functions of the mitochondria itself, specifically oxidative phosphorylation and ATP production. What are neurodegenerative disorders? As these are essentially disorders that result in excessive neural pruning and neuronal death. There aren't many cures for these. You can't take away the symptoms, but we can help keep them in check, and those are called therapeutic interventions. VRET, exposure therapy, music therapy, sound therapy, etc., etc., etc. Examples of neurodegenerative disorders include Alzheimer's, Parkinson's, epilepsy, dementia, etc. Mitochondria provides essential energy metabolism mechanisms for the formation of neural pathways. And this is linked to neurodegeneration because whenever there's an impairment of mtDNA that impairs the energy metabolism, that can also cause, by chain reaction, a lack of neural pathways. This is shown with this image right here. As you can see in the image, the ROS is whenever there's a mitochondria malfunction or mtDNA mutation, there's an increase in ROS, and this increases the oxidative stress of the mitochondria, therefore causing an increase in cell apoptosis as shown in the top right corner, as well as a decrease in mitochondrial stability. So the link between mitochondrial genetics and specific disorders, right? So let's look at Parkinson's disease. One of the many links that we've seen between Parkinson's disease is that the mutations to DNA polymerase gamma, which actually helps with the replication of mtDNA, causes the mtDNA not to be able to be coded correctly, to be replicated correctly, and this causes an impairment in the mitochondria function. And it's actually associated with Parkinsonism or Parkinson-like symptoms. The similar thing has been found in my studies, and how this time is with cytochrome C oxidase activity, and how this is linked to Parkinson-like symptoms. Huntington's disease is very similar in how you have a mutant, a Huntington protein, a mutated Huntington protein, which is binding to the outer membrane of the mitochondria and just causing damages with the energy metabolism. So, before in the previous slide, we looked at the correlative effects, right? How does the correlation between mitochondria, mtDNA, and neurodegenerative disorders, right? However, it's very important to understand that we need to look at causative effects, right? And Exia et al. Tried to find this causative effect by conducting a Mendelian randomization analysis. They just took a bunch of data, genes, and just did a mtDNA analysis, right? And when they did this, they found that mtDNA does link to neuronal death, but it doesn't have enough evidence to be a cause of neurodegenerative disorders. And essentially, this just calls for further research into mtDNAs and its function within the cell and how it can cause neurodegenerative disorders. Is whether it is a correlation or causation, we're not sure yet. This study kind of just shows the ambiguity there. So possible treatments. One possible treatment we have is obviously just adding new mitochondria. We call that mitochondrial transplantation, right? Another treatment is CRISPR, in which we, through a very tedious process, as shown in the image, with the CAS9 protein, along with the cutting and like the target sequence and a bunch of stuff like that, we are essentially just trying to code the SNCA protein to prevent synuclein buildup, which can accommodate for the increase in synuclein buildup caused by a mutation in mtDNA. So the outside DNA helps for the inside DNA. But instead, why don't we just attack the inside DNA? That's a very recent treatment called mitochondrial gene editing. And that's exactly what mtDNA or mitochondrial gene editing does. It attacks the mtDNA and tries to fix the inside DNA itself. So, societal implications. So understanding mtDNA can save so many lives and provide avenues for research unlike we've ever seen before. It can save millions of lives, but we have to understand that we need to evaluate the extent to which this is okay without causing long-term damage. How can we do this without hurting someone more than we're helping them? That calls for more research into what mtDNA is and how it helps or how it codes for neurodegenerative disorders. Thank you.